Welcome to the first session of our Reproducibility in Science webinar series. These days, the word reproducibility is being thrown around in industry literature, on advertisements, and even by journal publishers themselves. But the question is, why does it concern you? And what does data reproducibility have to do with getting published? And which factors affect your research? In this webinar series, we've set out to help you recognize why making sure your research is reproducible is important, and how conducting validation studies early on can help you succeed. This first session is all about the Reproducibility Initiative and validating experiments in independent labs. We have Dr. Elizabeth Iorns, one of the co-directors of the Reproducibility Initiative, with us today to tell you all about it. But I'd like to find out if you've heard about the initiative previously. So before we get to our guest speaker's presentation, let's find out what you know in this poll question. Okay, I've popped up the poll question for you to answer. The question is, which factors influence data reproducibility? Go ahead and submit all of the answers you believe apply. You can choose experimental design, choice of reagents, detection methods, documentation of protocols, or fraud. We will give everyone a few moments to enter their thoughts and then reveal the results. All right, here's what you think. Looks like you've covered them all, so that's good. During your talk, Dr. Iorns will tell you that it is indeed all of these things that affect reproducibility. But when it comes down to it, it is the ability of the entire experiment to be reproduced that is important. Dr. Iorns is the founder and CEO of Science Exchange, the co-director of the Reproducibility Initiative, and is a part-time partner at Y Combinator. Elizabeth has a PhD in cancer biology from the Institute of Cancer Research, and before starting Science Exchange in 2011, was an assistant professor at the University of Miami, where she remains an adjunct professor. Elizabeth has received a range of honors and recognitions, including the 2012 Kauffman Foundation Emerging Entrepreneur Award, one of Nature magazine's 10 People Who Mattered in 2012, and one of Wired's 50 Women Who Are Changing the World. Elizabeth is focused on the development of innovative models to promote the quality and efficiency of science research, and we are honored to have her as our guest speaker today. Let's hear what she has to say. Elizabeth?
Thank you very much, Ashley. I'm really happy to be here today to talk about creating a culture of reproducible research. So basically, what I want to talk about today is improving the quality of scientific research through independent validation studies. So reproducibility can be affected by many different factors, and we've studied a lot of them. Some of them include things like experimental design, the choice of reagents and detection methods used, proper documentation of protocols, and in very rare instances, fraud. So just to give a brief description of what reproducibility is, it's the ability for an entire experiment or study to be reproduced. And in particular, we're focusing on inter-laboratory testing. So we're looking at the ability to independently reproduce results in an independent lab. And reproducibility is actually fundamental to the scientific method. When we think about the scientific method, it's all about being able to repeat steps to obtain similar results. And fundamentally why reproducible research is so important and why we focus on it so much is that the ability to build effectively upon previous findings is based on the fact that they are reproducible. So one of the questions that brought the issue of reproducibility to light was whether or not published results are reproducible. And there's been some studies that have been published that have brought this into question. So in particular, this was a survey that was published from MD Anderson, who examined the academics' experience with reproducing published data. And they found that about 50% of respondents had experienced at least one episode of the inability to reproduce published data. So this result was fairly high, but comparable with other results that have subsequently been published about the reproducibility when replication studies were conducted. So there are very few replication studies that have been conducted, but of the ones that have been conducted, the results are fairly worrying. So the first is from the ALS Therapy Development Institute. They retested more than 70 drugs that have been published in 221 independent studies, and they were not able to reproduce any of the results. And in fact, some of the results had been shown to be effective in multiple different studies. For example, minocycline. This was shown to be effective in 4 separate ALS mouse studies, but in a clinical trial of more than 400 patients, it actually worsened symptoms. And when the ALS TDI had replicated this experiment independently, they failed to see an effect in their mouse studies. The second example is from the NINDS that replicated 12 spinal cord injury studies and fully reproduced 2 out of the 12 findings. Bayer and Amgen are the 2 most widely cited studies that have been published, and these were just summary data that indicated very low rates of reproducibility in their in-house target validation studies. So Bayer published that 14 out of 67 studies that they investigated, they could reproduce the results, and Amgen only 6 out of 53. So the data that we have so far suggests that the answer to whether published results are reproducible is that they are not always reproducible. People tend to not worry about this so much because they assume that the literature can correct itself over time. But actually, when you examine the data to support this presumption, it's actually shown to be incorrect. So there's very little self-correction of the literature. Only 0.2% of the literature is ever retracted, and this is usually reserved for cases of fraud, versus an approximation of around 70% irreproducibility. Very few researchers published negative findings. So of the researchers that were surveyed by MD Anderson, who indicated that they could not reproduce published findings, less than 30% of them published their failure to reproduce findings. And only 14% of the literature at all reports any negative results. So this suggests that we have a very strong positive publication bias. In addition, there is little evidence that additional publications from other academic labs are able to identify reproducible results. From the Bayer publication, they stated that we didn't see that a target is more likely to be validated if it was reported in 10 publications or in 2 publications. And an example from Pamela Ronald's lab at UC Davis, where she retracted studies published in PLOS and Science from her own lab when she realized that there was a reagent error. She stated that the results had been confirmed independently by 3 other groups, and yet nobody had published any negative findings associated with that finding that she published. So this suggests that the literature is actually not self-correcting. Another indicator of reproducibility, or at least quality, is citations. And this has been used as a basis for measuring the impact of published results. However, none of the replication studies reported any correlations with citations or a related indicator, journal impact factor. So the NINDS studies found no significant difference. Bayer saw no significant difference, and Amgen saw no significant difference between the citation rate of papers that were reproducible versus non-reproducible. So then much of the literature might be irreproducible, and our measures of quality, like citations, do not seem to indicate whether a result is reproducible. So that suggests that we have a problem. So what can we do about this problem? One of the things that we've been looking at is ways to change the culture, to create a culture that incentivizes the publication of reproducible results. And how we're doing that is to verify key results using independent replication studies. This is because independent replication studies identify robust reproducible research. So these are very valuable studies to conduct. Bayer found that either the results were reproducible and showed transferability into other models, or even a one-to-one reproduction of published procedures revealed inconsistencies. And so this suggests that doing replication studies directly provides a screening tool for identifying robust reproducible research that can be effectively built upon and transferred into other models. However, it's very difficult to incentivize replication studies. Some quotes from various different publishers. Forbes said pressure testing academic theories remains an important and profoundly undervalued activity. The Economist stated, nowadays verification studies do little to advance a researcher's career, and without verification, dubious findings live on to mislead. And Wiley study said that those who document the invalidity of a published piece of work seldom get a welcome from journals, funding agencies, and conference organizers. So this is also related to funding. Replication studies are very hard to fund. So Pamela Ronald, who was the investigator who retracted studies in PLOS and Science, she said that it's virtually impossible to obtain funding to repeat results that have already been published. So it was very difficult for her to determine whether she should retract her studies because she could not obtain funding to really do the experiments to repeat work that she had already published. But people recognize the importance of funding replication studies. Bruce Alberts mentioned this at a testimony at Congress where he talked about funding agencies and the importance of encouraging replications. So to provide an incentive system and a mechanism for supporting replication studies, we've worked on several different projects, and these include the Reproducibility Initiative. This is an initiative that aims to promote a culture of reproducibility by incentivizing, rewarding, and facilitating the conduction of high-quality independent replication. So the aim of the Reproducibility Initiative is to identify and reward high-quality reproducible research. One of the major studies that we have been involved in at Science Exchange is doing the Cancer Biology Reproducibility Project. This is a collaboration between Science Exchange and the Center for Open Science, and has been funded by the Laura and John Arnold Foundation and supported by numerous reagent donations. This project is independently replicating the key experimental results from the top 50 cancer biology studies published from 2010 to 2012. And you can learn more about this project at cost.io/cancerbiology. Studies were selected from 2010, 2011, and 2012 based on the number of citations for cancer biology search terms in Web of Science and Scopus. We also examined leading indicators of impact like Mendeley readers and altmetric.com data. We then excluded reviews, clinical trials, case studies, and sequencing papers to select 50 studies that had the biggest impact on cancer biology to be replicated by this project. So we are replicating the key experimental results that were published in these studies. So there's no initial exploratory work that's being conducted. We use the published results to calculate sample size needed for at least 80% power to detect the reported effects. And we're able to rapidly and cost-effectively replicate the studies using the technical experts in the Science Exchange network. And this has allowed the project to be conducted for $1.3 million. What happens is we do a high-quality replication. We use a direct replication using the same materials and methods as in the original study. We pre-register protocols in the Open Science Framework, and the studies are peer-reviewed in the journal eLife. We obtain input from the original author on the proposed replication protocol, use power calculations to ensure adequate sample size, include controls to show that the replication experiments worked, use expert independent labs from the Science Exchange network with extensive expertise in the techniques being replicated, and publish all protocols, results, and data in the Open Science Framework as well as in eLife for review by any interested party. So the labs doing this work are comprised of labs within the Science Exchange network, which represents a network of verified high-quality research labs that are available to conduct experiments on a fee-for-service basis. These labs represent labs from more than 400 different research institutes, including 75 of the top 100 recipients of NIH funding in the United States. So Science Exchange essentially provides a marketplace online for ordering experiments and promoting scientific collaboration. So it enables researchers anywhere to order experiments from the world's best labs. And LI-COR, who we're working with to conduct these studies, is one of the Science Exchange labs, and they provide the access to high-quality, reproducible Western blotting services available from their Science Exchange page. The goals of a cancer biology reproducibility project are to show that direct replication is possible in a cost-effective and scalable manner, as well as to provide a complete open data set of high-value replicated studies, and to develop reproducibility best practices. Overall, we want to create a culture of reproducibility. So you can track the progress of this project at cost.io/cancerbiology. Several additional reproducibility projects have been launched and are currently being funded, like the Neurodegeneration Reproducibility Project, to extend these types of high-quality replications into other fields. The ultimate goal is to align people's incentives to identify and reward high-quality reproducible research using independent replication studies as the mechanism to do so. This will allow us to identify reproducible research that can be quickly and cost-effectively built on. The Reproducibility Initiative is currently expanding its program to new fields and funders who wish to verify their results. Importantly, the infrastructure and methodology that's been used in the open replication studies is also available privately through the Independent Validation Service. And this service has been used by numerous pharmaceutical companies and funding agencies to validate key experimental results that they wish to build upon. One example is actually the Antibody Validation Project. This is a project to independently validate antibody reagents so that researchers can determine which antibodies will work when ordered and used in their own labs. Here's one example of a LI-COR antibody that was validated independently through this program. So in summary, there are many opportunities to improve the efficiency and quality of scientific research, and we're really happy to have worked on both of these initiatives with Science Exchange and the Reproducibility Initiative. So thank you very much for listening. I'm going to turn the presentation back to Ashley for closing.
Thank you for the informative talk, Elizabeth. I definitely agree that it's important to ensure that all scientific experiments, and especially published results, are repeatable. Incentivizing scientists to reproduce their research through the Reproducibility Initiative and Science Exchange, and providing antibody validation through Antibodies Online will only make the process easier. It's exciting to think about the kinds of research discoveries we will be able to make by building upon the sound science that researchers like you, our listeners, are conducting. The remainder of our webinar series is meant to help each of you continue to improve on the methods you are using. If you remember the poll question we asked, there are several factors that affect the reproducibility of research. In our next session, which will happen on April 15th, we will discuss the optimization of one of the most common cancer research assays, the Western blot, and how a few small changes in your protocol and reagent choices can mean significantly improved reproducibility and the best results possible. The final webinar will introduce you to the reproducibility problems associated with your choice in Western blotting detection methods, then present solutions for getting the best data possible every time. Lastly, stay tuned for an announcement this fall about new tools from LI-COR that will help make it easier to record and share your experimental data. Before we close, let's take one last poll. So, with the influence of the Reproducibility Initiative, the future of cancer research looks promising. What do you plan to do to ensure you are a participant in the culture of reproducibility? You can choose attend the next webinar, order an experiment from Science Exchange, share this information with my colleagues, or look at the changing publication guidelines online. We'll give everyone a moment to choose an answer or two, and then show you the results. Wonderful. It's great to know we have such a solid group of scientists forging ahead with robust research. Again, I'd like to thank Dr. Iorns for her time today, and all of you for participating in our discussion. If you would like to find out more about new publisher guidelines, visit our blog at www.licor.com/bio/blog. And watch for a replay of this webinar to be posted at www.licor.com/webinars. We look forward to seeing you at the next webinar. Right now, our panelists are still answering questions, so please stick around if you have a question that has not been answered, or feel free to give us your contact information if you'd rather receive an email response. We will be taking questions for a little while longer, so you may still submit any questions that you have. Lastly, please let us know how we can improve our webinars by answering the post-event survey questions when you exit this webinar. Thank you for joining us today, and we hope to see you soon.